the participants. Now I will hand over this session to our expert. Sir, please, sir.
Okay.
Yeah, thank you, sir.
Okay, so hope you remember some of the aspects we discussed yesterday. So could you tell something what we discussed yesterday? Secondary structures. Okay, so what are the some part of secondary structures as well as some features from the primary structure. Okay, so what are the regular secondary structures? Alpha helices and beta strands. So what are the major interactions for the formation of secondary structures? So what's the non-covalent interaction involved in alpha helices and beta strands? Okay, this is another secondary structure, fine. Okay, so what is the non-covalent interaction involved in the formation of secondary structures? For example, alpha helices or beta strands. What are the non-covalent interactions in protein structures? We discussed about several non-covalent interactions. These hydrophobic interactions, electrostatic interactions, van der Waals interactions, hydrogen interactions, and all. Right. So which interactions are okay? The hydrogen bond is the non-covalent interactions involved in the formation of secondary structures. Okay. Okay. So what is an alpha helix? So how alpha helix are formed? There is specific interaction between I and I plus 4, right? C1 NH atom, NH, right? And how many residues in one turn? In an alpha helix? 33.6 residues per turn, right? Okay. And yesterday we discussed about the propensity. So remember propensity, what is the propensity? Right? How to calculate the propensity of helices? Yeah, it is tendency of any amino acid residue to be in alpha helices or beta strands, right? So how to calculate the propensity? I discussed yesterday about the propensity. Yeah, I will do. First, give me the answers before that, because I like to know whether you are following or not. Okay. Yeah, the percentage of residues in alpha helix. Okay. Yeah, the percentage. Yeah. Right. So then we discussed about the some residues which are helix formers. Can you tell any one of the residues which prefer to be in alpha helix and residues which block alpha helices? Yeah, leucine, renin, glutamic acid, they prefer alpha helices, and the proline, glycine, right? So they are not preferring to be in alpha helices, right? And we discussed about the secondary structure prediction. What are the different secondary structure prediction algorithms we discussed? Yeah, statistical analysis, information theory, and multiple sequence alignment, machine learning, and consensus methods, right? And heterogeneity profiles. So we discussed various methods to predict the secondary structures. Okay. Okay. Today we'll move on to the tertiary structure. Okay. So what do you think about protein tertiary structure? Okay. So what is protein tertiary structure? Which information we get from protein 3D structures? Coordinates, right? So you'll get the exhibition coordinates of any of all the atoms, right? In a ST or in a protein, right? So it provides information regarding 3D structures and the location of each atom, right, in a protein, in a Cartesian coordinate system. So you can see here. Okay, so this is a tertiary structure, and you get the complete information regarding the protein 3D structures. Why the 3D structures are important? Why do we care too much about the 3D structures? 3D structures are important. But what are the applications of these 3D structures? Okay, so 3D structures, okay, it will tell you where we have the residues are located and what are the possible binding pockets, right? Even if in the COVID-19, if you want to develop any therapeutics or any drugs or vaccines, so they have to discuss with the targets. So they should know where are the binding pockets, where this small molecule will bind, and then how the mechanism of action and all. So it is very important to have the protein 3D structures to understand their functions. And that is the reason many investigators are spending time on protein 3D structures. Okay, so how to get the structures? Experimentally, how can we get the structures? There are various techniques like X-ray crystallography or NMR spectroscopy or cryo-electron microscopy, right? Various techniques, right, which are currently available in the literature, right? And many sophisticated instruments are also currently available, right, to determine the 3D structures of protein molecules. Okay, so we talk about, I don't want to give more details about the experimental technique, just as a brief, I'll tell you. Okay, the X-ray crystallography, it involves various steps. First one is we need the protein in a pure form. So because if the protein has impurities, then we don't get the proper structures. So it's a protein purification, but they like to purify the protein. And then the X-ray crystallography requires a crystal. So it's a periodic arrangement of atoms, right? So it's a crystal, then only we can get the diffraction and we can get the electron density map and we can get the structures. So this is the bottleneck for most of the cases. That is the reason why there are very less number of membrane proteins available in protein data bank, because the membrane proteins are very difficult to crystallize. Right. So second is the protein crystallization. Then you go to the crystal mounting, because mounting is very important because then you have the proper diffraction patterns, and then they will collect the data. What are the data they collect? They will collect the distribution of electrons of molecule, that is the diffraction patterns. That's called the electron density map. Then they get the electron density map, then they process the data, and then to identify where the atoms are located, and then what are could be the probable residues or atoms, and what the probable coordinates. Then we have the structure solution, refinement, and then we can use it for the analysis. There are various methods available to check the resolution of all these structures. One of the methods is Thompson plot, right? I discussed yesterday. So if it is the allowed region, then they'll think, okay, these structures are in good form. Well, this is one of the methods to determine 3D structure, the X-ray crystallography. So you can have the crystals here and this electron density map here. Finally, using the computational software, they can develop these structures. Right? This is a static structure, right? But if you want to have dynamic structures, you can do the MD simulations, so you can create a lot of dynamic structures for any of your proteins. And the second technique, okay, this is the NMR spectroscopy. It is based on the spin, quantum mechanical properties of the atoms, like the spin of the spin, and determines information about the atoms, right, with respect to the magnetic fields. So you can spin of the system, so you can get the information regarding the atomic positions. Based on that, they will develop models. That is the reason if they have the NMR spectroscopy, you can see multiple models in the protein data bank, right? Because they will develop several models based on the spin of the system. Okay, so all these structures which are solved at atomic resolution, or using an X-ray crystallography or NMR spectroscopy, or you can do the cryo-electron, the recently developed technique. So all the deposits in the protein data bank. Protein data bank is the unique resource for protein 3D structures. What is the resource for protein sequence? What's the database of UniProt, right? So how many? Okay, correct. So how many data in UniProt database? 180 million, right? Correct. So do you expect what do you expect? How many structures are available in protein data bank? I will just in a few minutes I'll give you the statistics. How many structures do you expect are available in protein data bank compared with UniProt? UniProt has 189 million sequences, right? So how many structures do you expect in the protein data bank? Approximately? Any numbers? 50 million? If 50 million structures are available, then we can get a lot of information regarding this. So it is very less compared. That is 1 by 1000 of these sequences, right? Now I'll tell you exact numbers now. Okay, so there's 1 by 1000 of the sequences available in the UniProt, right? So why this X-ray crystallograph is very important? Why the structures are very important? Because these structures are helpful to understand the functions. And if you see, I show some of the list of the Nobel Prize. Okay, the people who got Nobel Prize just by solving the structures. Starting from the 1962, Perutz and Kentru, right? They solved the structures of the globular proteins like the myoglobin and hemoglobin, right? So to understand the secondary structure, that is the reason why they got the alpha helices and beta strands there. But mainly the globins are having alpha helices, right? And they solved the structures of these globins and they got the Nobel Prize in 1962 for chemistry. Then 1962, they did the Watson-Crick, they got the Nobel Prize for the DNA. And Dorothy Hodgkin for the vitamin B12 complex, they solved the structures to understand the structural importance of the biochemical substances like vitamin B12. So she got the Nobel Prize in 1964 in chemistry. Followed by that, 1972, and Finson. So he showed that the information which have the structures contain are available in the sequence. That means if you have the amino acid sequence contains information regarding the structures, and this particular sequence can hold in one particular form in the 3D structures. It cannot take any random confirmation. So this is what he got the prize, and he did the experiment with the ribonuclease, right? And he showed that amino acid sequence contain the information regarding the active conformation of the particular protein. Then 82, Aaron Gluck, he solved the success of protein nucleic acid complexes to understand the importance of the protein nucleic acid complexes. As I mentioned yesterday, 1988, right? Eisenhower, Rubert, and Hermit. So they got Nobel Prize for solving the structures of membrane proteins. At the first membrane protein, photosynthetic reaction center, they solved in 1988. So to understand the mechanism of the photosynthesis. And in 91, Ernest got the Nobel Prize for NMR because he wanted to use NMR, right, for the protein structures determination. And followed by that, Kurt Udrich, 2002, he extensively used NMR to solve the structures of proteins, and he got Nobel Prize in 2002 in chemistry. Then he got Nobel Prize for the ion channels and the eukaryotic transcription, and Venki Ramakrishnan and the group in 2009 got the Nobel Prize for the ribosomes. That time it was very popular at that time, 10 years ago. In 2012, for G protein coupled receptors, so the Kobilka, he got Nobel Prize in 2012, followed by Martin Karplus and Levitt. So they got Nobel Prize for the MD simulations. Because all these structures are static, so if you want to have the dynamical structures, you can use MD simulations. And say for the MD simulation models, they got Nobel Prize in 2013. And 2017, so the cryo-electron they developed, so to understand the high-resolution structures of the biomolecules in solution, right? So they got Nobel Prize in 2017. Right? That means the structures are very important, and the technologies as well as the structures of the proteins, right? Have any unique structures, right? So these people, they got the Nobel Prize on various aspects. Okay, so now all the structures are deposited in protein data bank. Protein data bank has 3 different places that are maintained in USA, that is San Diego Supercomputing Center, and PDB Europe, and EMBL, as well as PDB Japan in the Osaka University. So they maintain this PDB. So then organizers, they act as deposition, data processing, and the distribution centers on the PDB data. Right. So recently they developed. Earlier they started 1977. The Brookhaven National Laboratory. They started the structure, the data acquisition with less number of structures, and the research collaboratory for structural bioinformatics. They developed a consortium to collect the structures and they deposit, they accumulate and maintain this database. Right. So another aspect is now, as of now, if you want to publish any article related with any of these 3D structures, you have to get the PDB code from the protein data bank. Unless you have the code, you cannot able to publish your paper in any of the journals, leading journals. So if you publish any of the leading journals, they will ask for the PDB ID from this protein data bank. That is another reason why the growth is very high. And the crystallization is very difficult, and getting the structures is very important, and also it takes a lot of time to solve the structures. That is the reason even with very good sophisticated instruments, the number of data in the protein data bank is very less. Okay, now I give statistics. As of today, okay, the number of structures available in PDB is 169,681. Right, compared with the UniProt sequences, it is 189 million sequences. It is 1,000 of the sequences available in UniProt. So this is the current status. That's 169,000 sequences, right, in the protein data bank. Fine, right. So now if you see the distribution of these structures in protein data bank, okay, you can see the X-ray crystallography. You can see the maximum 150,000, followed by NMR only 13,000, and EM 6,000. And we have more number of data for the proteins. So you can see the proteins, you can see 148,000, followed by the nucleic acid is around 8,000, right? And the only nucleic acid is around 3,500. Okay, this is the current statistics of the protein data bank. Okay, it's available in the www, right? This is website, rcsb.org. So you can go there and you'll get all the information, right? So recently, if you see, they developed another link for the coronavirus resources. So you can see the COVID-19 coronavirus resources, right? They made together all the structures related to coronavirus resources in the protein data bank. Okay, we see that. Okay, this is the first solid structures of the related to coronavirus, right? Because 6LE7 is the main protease with the inhibitor. Okay, you can see the inhibitor here, and this is your main protease. Right. Then here, this is the structures with the SARS-CoV-2 and the SARS main protease that happened in 2004. Right. So you can see how they are very similar. They're about around 80% similarity, right? And you can see the structural similarity between the SARS and SARS-CoV-19. Right. This is the structures. And here you can see the dimers, the interface. Okay. Here you can see the ACE2 enzyme, right? As well as the spike protein, here is spike protein and enzyme, and here you can see how the interactions, right? So this is how they pass the information to the cell, and it having this affect the human body, right? So to tackle these things, there are various studies this is going on to develop vaccines or develop any therapeutics or the drugs. And to understand the mechanism, you can see every day, you can see so many nature and science papers, they discuss about the vaccines or antibodies or the small molecule therapy and so on. So we also did analysis, maybe we will publish in maybe sometime in March. So already these papers still in press, but we got 150 citations so far within the short period in 3 months. We tried to understand the synergism effect of this molecule, these drugs. So we studied the drugs used for HIV as well as the flu, and see how far these molecules interact. Because if you see in these structures, the protease, these 3 molecules, they have 3 different binding pockets. One is here, and one is here. That is why there is a synergism effect of these molecules. It increases and enhances the binding affinity as well as this interaction energy. So we could study the synergy effect of these compounds to this main proteins. Fine. So now if you go to the PDB, right, okay, I'll go a little more about the PDB, right. So if you take one protein here, but this is to Elisum, it is lysozyme, right. So you can have the various options where you can have the sequence information or the structure information, or you can have the PDF file and so on. If you click on the FASTA sequence, right, this will give you a sequence. Okay, this is the one we discussed yesterday and day before yesterday. So 3D structures here also you can get the information regarding the sequence. Okay, this is the sequence information for a particular protein, right? So then what are the contents of the protein data bank? What are the information you can get from the protein data bank? I'll show some of the details, and when you have time, you can go through the details, right? It's available for free to everyone, so you can go through details and get more information. Fine, so it contains the source and it contains the name of the protein. This is T4 lysozyme, and here the organism is phage T4, right? And where they published, right? And then you can see the resolution and the sequence. This is your amino acid sequence shown here, and this is your secondary structures, right? This contains both helices and strands, and the helices. This is the details of the helices, and here your details of the strands, right? And this will give you the atomic coordinates. So you can see here the coordinates of all the atoms and residues, right? And here, if there are any water molecules, they show this as the heteroatoms, right? Fine. So in the x-y-z coordinates, okay, here I showed that this is x, this is y, this is z coordinate, and after that we have this another line that is the occupancy, and this is the temperature factor. What is the occupancy? Because sometimes they make different confirmation. If this could not be able to fit in the rectangle density map, and they take the different occupancy, maybe they get two different confirmation. One has the possibility of 0.57, this has the possibility of 0.43. In any case, total will be 1. So in this most of the cases, if it is 1, that means it is only one position, so that will fit in the rectangle density map. So this is equal to 1. If it is not fit, you have two confirmations, right? So then you can see the anisotropy, you can see the two values for each atom. So total will be 1. That is the called occupancy. And the second aspect, that is called temperature factor. Temperature factor will tell you how far these atoms are flexible, or it is rigid, or how far their vibrations. Whether there is fixed within the rectangle density map, or it couldn't fix it. So if you see some of the residues of the atoms, you can the values are very less. That means it is very rigid. So they are well packed. And if some cases if it is very high, that is more than 50, these are flexible. They are much more oscillations. And here if you see most of them are blue. Blue are good. There are less fluctuations. And some of them are in red, and some of them are in yellow. They are having the high fluctuations compared with the other atoms. Right? That is called the temperature factors. When we go through a sequence, so if you give a sequence here, you can see a sequence, and if you have some structures, right? Some of them are spiral, some of them are arrow. So what is the meaning of the spirals? Spirals tell you the region where they have the helical regions. Okay, this is the helix, and here you have the helix, here you have the helix, helix, right? So what is a helical segment? First helical segment. Can you tell me a helical segment that starting from this I to this E? That is a helix, right? Okay, this is the first strand. Okay, this is the second strand. This is the third strand, and this is your bend or the turn. And here again, next helices, and this is a very small segment of beta strand and so on. And this you can get this information from protein data bank. Okay, and then you can have this the classifications, whether this proteins contain mainly predominantly alpha helices, or it is with the beta strands, or it is a combination of both helices and strands. So if it has both helices and strands, they call alpha plus beta or alpha with beta, depending on the arrangement, whether the helices and strands are separated or they mix each other. If you see here, most of them here completely helices, and this region is strand, so they are separated, but contains alpha helix and beta strands. This is why they classify as alpha plus beta by using this curve. In the cat, they consider the contents of the helices. That is the reason they classify this as mainly alpha in the case of the cat classification. Then go to similarity. So this will tell you how far this protein is similar to other proteins, right? So this is other codes where the similarity is very high, because yesterday we discussed the UniProt regarding 50%, 90%, 100% sequence similarity. Here we have various similarities, 30, 40, 50, and so on. Right? If you want to have it, you can get the similar sequences for your given sequence. And we have the 3D structure similarity, because we have the sequence similarity, likewise the structures, right? I showed here. Okay, so this is your structure similarity you will get. Okay, if you superimpose two structures and how far they are structured similar. So you can calculate using the root mean square deviation. And so here this is the structurally similar proteins with the deviation is very less, for example 1.29 or 0.4 and so on. And then the literature, this is the papers which describe this specific protein, and what are the papers which cited this PDB, cited this 3D structures. These are the proteins cited protein data bank. Okay, this is my paper. So they listed my paper also that's a citation of this PDB. So these are the structures we use in our analysis. That is the reason why they listed my paper also in this protein data bank. Then they given the information regarding the chemistry and the biology, depending upon the active sites or the crystals, the unit cell parameters, or the refinement, and so on. Okay. So now this is the bond length, bond angle, and torsion angle. If it's the bond length, for example, if it's C and N, so this is the minimum bond length, and this is the maximum, and here you have the standard deviation for this bond length. Right. Here you have the bond angle. Right, so here is the angle, this is the minimum angle, this is the maximum angle. So you can have this the average angle, and this is your deviation for any of these angles. Right, then you have diagonal angles. Right, how many atoms are required for to form diagonal angle? 4, right? Yeah, we discussed very clearly yesterday. So we have that you can see the diagonal angles for phi angle, psi angle, and chi angle, and omega 1, omega 2. So this is your minimum angle, is your maximum angle. This is your average as well as the deviation for the different amino acids. Well, then again, it has the links to other databases, right, and so on. Then if you go to advanced search, so you can search based on the different aspects, the ID or the structures or the molecular type. If you want to do the molecular type, if you want to have it protein DNA complexes containing proteins, yes, because we want to have a protein DNA complex. And the DNA, yes, RNA, no, hybrid, no. If you do this, then it will ask for the identity, if you want to remove the identity or not. If yes, then you click on here, and they ask for which range you want, with 40%, then this will give you all the details. Okay, these are the number of citations, these are the number of structures, and this is the different classification based on organism or taxonomy or methods or scope or research and so on. Right? Fine. So now we have the structures, and you can play around the structures using different visualization tools. Right? So there are various visualization tools available in the literature, like PyMol, RASMol, JMol, King, WebMol, and SpacePDB. And PyMol is one of the most widely used software, and it is earlier it was completely free for everyone. And due to the advancements in this PyMol, so Schrodinger bought PyMol, and then they incorporated this into the commercial version. However, for the limited options, it's available for free for educational purposes. Right. This afternoon, I will give you some questions to use the PyMol, and we can use PyMol extensively, and I will show you how to calculate various aspects using PyMol. That will be interesting. I will show you it. Right. Okay, so what are the different experimental techniques to solve the structures of 3D structures of proteins? X-ray crystallography, NMR spectroscopy, and electron microscopy, right? So totally now, how many structures are available in protein data bank approximately? Good. So how many structures available in protein data bank? Around 116,000, right? These many thousand structures available. So what are the various information available in the protein data bank? Coordinates, temperature factor, occupancy, along with other details about the literature, secondary structures, and so on. Right? All the information available in the protein data bank. Okay. So there are various visualization tools. I'll take the PyMol. Okay. So this is the registration page for PyMol. I encourage you to install PyMol for the afternoon class, because afternoon we have mainly the not the demonstration, as well as we'll ask you to do in your own computer, right? For this case, you need to install PyMol. Okay, go to this website. This is the registered installation website, https pymol.org, right? Right, if you go, just if you Google it, you will get the PyMol. Google with PyMol, you will get the site, and here you have to fill your details. And if you continue, right, you will get this the permission. So you can download and you can install the PyMol in your computer for the today afternoon's session. Right. If you don't have this install this one, then we can show the demo. But in this case, you cannot do in your computer. So that is a problem. If you do your computer, you can learn a lot, because here this is very widely used for many publication purposes, and you can calculate various parameters, and you can view the protein molecules in a nice way and all. Right? I'll show you a few slides to demonstration. I'll do it now. In the afternoon, we'll exactly do on your computer, so that you can practice. I prepared already about 25 questions, so you can answer all the questions. You can do it from your computer. Take note down again. This is the site pymol.org, and just you download it and install it and get ready for the afternoon's class. Okay. Okay. So when you click the PyMol, because when you install PyMol, it will open two windows. Okay, this is window number 1, this is window number 2. Right here, this window is a command prompt and menu options. You can see it's a command prompt here, and you can use any of the command prompts, right? So, and you can get any information from here. And this one is a viewer window, so you can see all the results here, and you can also play around with this window, right, to see the structures and to get a lot of information. Just I will explain some of these aspects. Okay, here first what we will do is we need to have the protein structure to view it, right? So we have to download any of these PDB structures which are from Protein Data Bank. Go to the Protein Data Bank RCSB website, and you select any of the structures and download one structure in your desktop computer, right? We'll also show you to download some few examples so that you can practice the same. So if it is there in your desktop, then open. So if you go to the open, right, so then it asks you which directory you have. So here 1TM is here, so you click here, and then we can see the structures. You can see beautiful structures. You can see very nicely the structure here. And usually a default view is they show a lot of lines, right? So can you see this structure? It is very complicated, right? You don't see anything like here. Right? Can you tell me how many helices and strands in this protein from this structure? No, you cannot say, because it is completely a line and wire model, so you cannot do anything from here. So I'll tell you in a few seconds. I'll tell you how many helices and strands, because if you change the mode, you can get it. So in the upside, if you say the top, you can say there are 5 buttons here. A is for the action, S is to show, H is to hide, L is to label, and C is to color. Okay, so I'll also explain the details of all these things. So A for action, S for show, H for hide, L for label, and C for color. Right? If you see this as the wire model, if you want to show us the cartoon, so now you can see the cartoon. Now can you see what are secondary structures available here? Approximately how many helices, how many strands? Okay, first question is how many molecules here? Is 1 molecule or 2 molecules? 1? I'll show you very soon. Okay, how many? Can you see a secondary 7 helices strands? Okay, fine, good. Okay, now if it's color by chain, because if you let's see, actually it has 2 chains. If you color by chains, okay, it is here color by chain, color. Go to C as a color and by chain. And if you color it, and you can see these are 2 chains. This one is here, one is here. Both are the homodimers, and this is the chain 1. This is chain 2. Right now it is an equilateral. When you color it, you can easily say look at two chains in this protein. Fine. So now if you hide one chain, because if you want to do some analysis to one chain, so you can hide chain B. So right click on the chain, go to that chain and right click, and you can hide everything. So you'll get that chain, and you'll get only one now. Right. With afternoon, you can do it on your computer, so don't worry too much. Just today, now I'll just show you a demo. Right, so you can hide this only one chain. So now you can see different secondary structures you can color. For example, helix, sheet, and loop. So they're asking which type of coloring pattern you need. So if you choose the coloring pattern, now you can see that, okay, this is loop, and red is your color, helix, and this yellow is your beta strand. So you can easily see the different types of secondary structures. You can also use different types of coloring patterns, so and you can get the different colors, right? This is helix, you can see this is stray sheet, and here this is your loop. Now, if you want to see a sequence, for example, now you have a structure. We don't know the sequence now. If you want to go to sequence, just you go to sequence, and if you display here, and you can see the sequence. And if you see the colors, these colors match with these structures. Okay, here also this is a strand, and here this is your loop, and here this is your helix. Right? This is what as I showed in the PDB when you discussed about the protein data bank. I showed the same secondary structures. You can see it here. Right? So it's a color scheme. Okay, so you can follow this scheme here. And this is your helix, and here this is your sheet, and this is your loop, right? And display the sequence with the color scheme, right? For example, if you take this region, this is some segments, right? Segments 21 to 26, right? So click on the sequence, right, or the structure. Okay, this is your sequence, and here is your structure, right? So now you can save it here, and it is here. The dots come here, and now we can expand. This is only the place where we highlight in the structure or the sequence. It is there. You can zoom it and the selected residues. So now you can get the zoomed residues, but we don't know which residue is what, right? So in this case, you can show in the case on the ball and stick. Now you can easily see that which atoms are carbon, which are nitrogen, which are oxygen. This is the convention used, right, in the literature. Carbons C, that is in green, and the nitrogen in blue here, and the oxygen in O. So this is O. Now you can easily see that, okay, here which are the residues based on your side chains, right? Fine. So now if you see here, if you want to label the residue, this is glue 23. 23 is here. Glutamic acid is E. It is the 23 is here, right? Oh, sorry. Okay. So it is here. This is hidden because you have the ring here. Fine. Now select this leucine, and you can label it. You can also label all the atoms: N, C alpha, C O, and C beta, C gamma, C delta 1, C delta 2. So you can label with any of these. These are the backbone atoms, N, C alpha, C O, and here these are side chain atoms. Right? You can label it, and you can see much more details. Then you can show this as lines, and also you can measure. You can do a lot of measurements here. For example, if you want to measure the bond angles, you can measure the bond distance and the dihedral angles. Right? How many atoms are required to have a bond length? 2, right? Fine. So now we can calculate the distance, right? So it's asking for the atoms. Okay, this is your first atom, CD1. This is second atom here, CG, and here this is the distance. They mentioned the distance as 1.5 angstrom, right? It is good. You can get the distance displayed in angstrom unit, right? That's 10 power minus 10 meter. Okay. So how many atoms are required for a bond angle? Yeah. Afternoon we have the practicals. At that time we will show everything. Just install your PyMol, and afternoon we will have the session, and completely you can do the practical online mode, and you can use that. As well as these slides are available in my NPTEL anyway. So they measure the angle for 3 atoms, right? You can see the angle mode, right? This angle is here. So go to angle mode. So if you select the atoms, right, you can, this is the first atom CD1, and second atom is CG, and third atom CD2. So here this angle, your angle is 109.9 degrees. So you can, it's 3 atoms, you can see the angle is 109.9 degrees. So easily you can do it. Then you can also see B factor just discussed. So you can display it, and you can see the show a surface. You can see which are the depends the colors. You can show a surface, and you can be the least flexible. I showed earlier, so it's a completely least flexible. And here this is the orange is highly flexible. Few atoms are highly flexible, which are outside, and the blue are completely inside. They are completely rigid. Right, so you can have the various option features for the PyMol. Okay, I'll discuss in the afternoon about the most of the features. Right, and this is tutorials and support they'll give in the PyMol page. Right, so there are various visualization softwares. Right, so now PyMol is the commonly used software, and you can use very beautiful pictures, and you can do a lot of analysis using PyMol. and afternoon we'll get the more details on the 5 more. So get ready with the installed 5 more version. Okay, so now the question is, okay, now like yesterday, I'll try to do some calculations, some mathematics I'll do. Okay, so if you go 3D structures, what can we do? Why you are so bothered about that? And what are the various parameters you can derive from these protein 3D structures and all? Okay, you can get all the information from my book. Okay, I showed the first class about my books. If you look at my books, you'll get almost all the information. And today I discussed only few aspects, how to calculate some of the aspects, where the contact maps, acceptable surface area, contact order, long range order, and surrounding heterogeneity. Okay, I think depending upon the time, I will cover some of the aspects. Okay, so what is a contact map? Contact map will tell you how far any two residues are in contact in space. In space, I mean the 3D structures. Right? There are in protein sequence, there are very, for example, 100 residues. Right? In sequence, sometimes they are very close, sometimes they are far. And how far these residues, which are close in space, either the residues are far apart in the sequence or they are close in the sequence, how they are close in the space. Okay. So in this case, we need to calculate the contact, right? And for this case, we need to see if the residues A and B are 1 and 2 are in contact or not. So in this case, we need to define few aspects. One aspect is we need to define a threshold. That means a distance. You can tell, okay, 8 angstrom or 6 angstrom or 5 angstrom. And the second aspect is we need to define the atoms from where to what to calculate the distance. One aspect is take C alpha atoms and get the distance between the C alpha atoms. Another aspect is you take any heavy atoms and do from residue 1 and any heavy atoms from residue 2 and see if any of the heavy atoms are within any particular distance, for example, 5 angstrom or 6 angstrom and so on. Right? Fine. So here I put the map. Here this x-axis is the residue number and y-axis is also residue number. If residues 1 and 2 are in contact, put a dot. If 1 and 3 are in contact, put a dot. Right? So finally, if you do that, you can get the contacts map like this. In this map will tell you, can you tell, see this map? There are why so many the dark at the diagonal. You can see the diagonal is very dark, right? What is the reason for that lot of dots at the diagonal and close to the diagonal? Any answer for that? Right? Yeah, because no, okay. So I'll tell you, okay. Because if you take these are the contacts. The contacts means the residue 2 and 3 in contact, right? So then you can see you can put a dot there, right? So you here close to diagonals means residue 2 and 3 are in contact or 2 and 4 are in contact because the residues are always in contact. The neighboring residues are always in contact. Either for each residue, both the directions, right? From the left side or the right side or N-terminal direction or C-side direction, they are in contact. Take for example residue number 10. 10 has contact with 9, 8, 11, 12. Always there, the 4 residues almost in contact. And plus they have further more contacts. These contacts you can see either if it comes from far from the sequence, there is dot here, or it is just nearby the sequence, it is dot here. But the one which are near the diagonal, that is the adjacent residues, the neighboring next residues. That is the reason why here you can find a dark dots here at the diagonal. Fine. So now if you, I'll tell you one question, so you can derive a map, then easily you know how to do it and why there are the contacts are the diagonals. Okay, this is the 3D structures. Because the XYZ coordinates are here. Okay, so I take only the C alpha. From this, I take the C alpha coordinates. I have the 10 different residues, XYZ coordinates. Can you construct a contact map? Put an x-axis and y-axis. and the x-axis number from 1 to 10, y-axis number from 1 to 10. And if 1 and 2 are in contact, calculate the distance. If the distance is less than 8 angstrom, put a dot. Likewise, 1 and 3. If this is less than 8 angstrom, put a dot. Can you make some dots and show me your picture? Right, x-axis 1 to 10, y-axis 1 to 10. If 1 and 2 are in contact, that means you take the calculate the distance, right, between 1 and 2, that x minus x bar whole square plus y2 minus y1 whole square plus z2 minus z1 whole square, and the distance is less than 8 angstrom, put a dot. Can you make few dots and show to me? This is how to construct a contact map. Anybody did that? If anybody is done, just show me in your video. That will be helpful for the others also. Anybody completed the work? Okay. So my question is, if 1 and 2 are in contact or not? Yes or no? If we take the 1 and 2, they are in contact or not? If you calculate the distance, whether the distance is less than 8 angstrom or not? Yes? I don't need the exact number. So whether what is the approximate number distance between 1 and 2? Are you able to calculate the distance? 60, not 60, not 60, because we have the square root of x1 minus x2 to the whole square plus y2 minus y1 to the whole square plus z1 minus z2 to the whole square. Here it is around 2, 2 square equal to 4. Here again 2, 4 plus 4, 8. Here again 2, 12. So square root of 12, that comes around 3 point something, around less than 4. Okay, fine, yeah, right. So that is less than 8. So you can get a dot, right? So if you make a graph, so 1 and 2 will get a dot. And how about 1 and 3? 1 and 3 is, what is the distance between 1 and 3? One and 3 also, if you see 36 minus 34, that is 2. 2 multiplied by 2 equal to 4. Here 17 and 23, that is 6. 6 and 6 is 36. 36 plus 4 equal to 40. Here around 3 and 49. So 49, square root of 49 is less than 8, so you will get a dot. Then if you take the 1 and 10, another example, take 1 and 10. So 36 minus 7, that is already 49. And 12 and 23, that is 12, that is 144. That's anyway, it's more than 64, right? So here they are in contact. 1 and 4, likewise, you have to do it, all combinations, right? If you do 1 and 2, no need to do 2 and 1, because that's same, right? then you can get the map. If you see the map, then you can see the diagonals, close to diagonals, you can see the dots, and you can see what are the other dots from the plot. Right? This is the contact map. Okay, so now you can see these contacts, and here if you see, this is the central residue. For each residue, you can see what are the residues which are in contact. Right? This is your sequence, and here this is your structure. And some cases, in the middle residue is T, right? Some residues which are close here, they are also here. And some which are far away, I did not include all the residues because it will be very crowded. So some residues which are far away, for example, it is here, this A is here. In the sequence, it is very far, but the structure, they are very close, right? This is the concept of inter-residue contacts, whether the two residues are in contact in space, but they are very close or far away in the sequence. Based on that, we define the different contacts, such as short range contacts and the medium range contacts and long range contacts. Short range contacts will tell you the distance is less than plus or minus 3 residues in sequence, and medium range contacts will tell you the plus or minus 3 or plus or minus 4, and the long range contacts will tell you it is more than plus or minus 4 residues. Right? So if you see here, the central one is T152, right? 152 and 153 are very close, they are short range. 151 and 154 are again within 2 residues, they are short range. And 152 and 156 are 4 residues, they are medium range. I put M here. And if you see 150 to 98, how many residues far apart? 152 and 98. How many residues far away in the sequence? If the one is 98 and another is 152. Right, how many distance far? What is the distance? The distance between 152 and 98? 50 residues, right? Yeah, 52 plus 2, 54 residues far apart, right? Right. Yeah, 54 residues far. So this, in this case, these two residues contact make which type of contact? Short range or medium range or long range? Long range contact. Right? Can you tell one more example to have a long range contact from this figure? Which two residues make long range contact? Another one example. Can you tell me a pair of residues which can form long range contact? (96) and 160. Why is 96? Yeah, 160 is here. 96, 95, 94. I think for 96 here. Okay, so this form 98 and 160, right? 98 and 160, this form long range contact. Can you tell one example for medium range contact? Yeah, 98 and 160, right? This two for long range contact, right? Can you tell one medium range contact between any pair? That means the distance should be plus or minus 3 or plus or minus 4. 153, correct, good. So can you tell one short range contact? That's within 3 residues, within less than 3 residues, to neighboring residues, right? So you can see, can you tell one example? 153, not the same residue, because these two, 153 and 153 are the same. So you should go for the another residue. All right, 153 and 154, that is short range contacts, right? So you can have the different types of contacts, right? Fine. Okay, now if you see the short range contacts only in the diagonals, medium range contacts are very close to diagonals, mainly in the alpha helices. If you see I and I plus 4, so they form the helical structures, so they mainly in the accommodated in the close to diagonals, and the long range contacts which are very far away, mainly they form the beta strands, because they have very parallel beta strand. You can see the distance, the residues are very far away in the sequence, but they are close in the space. So here you can see this mainly for the beta strands. Right? This is what I put the beta strand here, alpha helices here, and here is the close contacts, right? Fine, this is one aspect. Now the second aspect is, okay, solvent accessibility. This will tell you how far each residue in a protein is accessible to solvent. That means if you take a classroom, right? So many students are inside the classroom, who are very closely accessible to the teacher, right? For example, who are sitting at the outside the ring, right? They are highly accessible. For example, the outside the ring, right? Compared with those who are inside the ring. So here we use a probe of water molecule. You roll on the surface of the protein and see how far this can accessible and penetrate to the van der Waals surface of each atom. This will give you a solvent accessibility. So this I'll skip that one. And here I show the results. You can roll on the water molecule and see how far each residue is accessible. This is your accessible surface area. This is your amino acid residue, right? Can you tell which residues are highly accessible and which residues are completely not accessible? If the values are very high, that means the residues are highly accessible. The values are less, that means they are buried in the interior, they are not accessible. Can you tell one example for exposed residue? Here, this is the accessible surface area. Here, this is your residue. E, which number? Okay, here, right? The E4. So this is 146. Right? This is completely exposed because this value is very high. Can you tell one example for buried? Yeah, right. Leucine, so there is this leucine is 1 and salicylic is 0, right? So this is completely buried. So now we have large number of data like this. So for the computer scientists, they like to have these numbers. They can easily parse this number. But for the biologists, okay, they find it very difficult to deal with the numbers. If I show this in the form of pictures, they'll be very happy, right? So we developed a software called ASAView, right, to show the actual surface area of each residues in the form of figures. Okay, so here you can see the picture. Can you see the picture? What are different? Can you see the colors? Can you guess something about the colors and the size? Right? Okay, the colors will tell you the different types of amino acids. Okay, so if you see the gray is for the hydrophobic residues, and the blue one, can you tell what is the, okay, is it visible for you? Blue is for the positive charge and the red is for the negative charge. And if you see all the negative charge residues, positive charge residues, the polar residues occupy at the surface. Because like to interact with the other molecules, right, other ligands, right, having the interactions. And the hydrophobic residues, mainly if you see the inner core, completely hydrophobic residues, this is because they are completely hydrophobic, they form the core to make for the stability of the particular protein. One aspect. And the second one, if you see the size is different. So small size means it is completely buried, that is very less accessible surface area. And the large circles will tell you the high area, that is the reason why they are completely at the surface of this particular protein. Right? So you can tell from this figure, right? Can you tell which residues are the surface? Can you tell one example? You can see the K32 or E2, these residues are kindly at the surface, and you can easily, right, you can see the plot and which residues are surface and which residues are in the interior of the protein. If you see this number, it is difficult to find all the information. Okay, now I give you the regular plot, right? So you can see some of them are very high, some of them are very low. So here the different colors we use for the different types of amino acids. Okay, now we developed a server. It is good for the scientific community that they don't have to calculate and do all these things. Just key in the PDB ID and to get plots. So now you can see this the options what they give. And if you want a spiral plot, you will get the spiral plot. And if you get the bar chart, you will get the bar chart, and so on. And if you want more details, you can get the values, right? We will provide the values also for each residue. That is fine. You will skip. Okay. So what are different types of contacts we discussed? 3 different types of contacts we discussed. What are 3 different types of contacts we discussed? Short range contacts, yes. Medium range contacts, long range contacts, right? How to define long range contacts? Yeah, they are very close in space, but they are far away in the sequence. But why should the cutoff of 4 residues? Sometimes we can use the more cutoff we can use to define the long range contacts. Right? How to construct a contact map? What are the information required to construct a contact map? Which information are required to construct a contact map? Right, you need the 3D structure information, so we need the distance, you have the cutoff, distance cutoff, and you need to have the atom level, atom information, whether it is between C alpha atoms or between the center of centroid or between any heavy atoms. Right, if you have it, then you can make this. So, what is the meaning of this? The dots near the diagonal. That means they are very close in the sequence. That is adjacent or neighboring residues, right, in the sequence, right? This one. Okay. So what is the concept of solvent accessibility? Right. The accessible surface area for the surface residues, accessible surface area is very high or low. It's high, right? For the completely exposed residues, they have the accessible surface area very high, and the buried residues, they are completely interior of the protein, so they have less ASA values. Okay, I'll discuss few more concepts, right? You can calculate. So one is a contact order, right? Here, this will give you the relative importance of local and non-local contacts, right? How far these residues, right, which are located in protein space. So it is defined as CO, there is a contact order, which is given as sigma Sij divided by L into N, where Sij is the sequence separation. Right? That means if this T1 and T7, right, they are separated by how many residues? What is the distance of separation between T1 and F7? Yes, what is the distance between these two residues, T1 and F7? 6, right? Then the T1 and the D2, it is 1. T1 and L3, it is 2. So we can calculate the total sigma Sij. So can you calculate the delta Sij for all the residues? Because T1 and D2, T1 and L3, T1 R4, T1 T28, T1 A15, F7, V6 and G8. Right? What is the total delta Sij? Can you quickly calculate and tell? You have to do it with the T1. We have to calculate with all the residues 1, 2, 3, 4, 5, 6, 7, 8 residues and take the sum. Okay, T1 and D2. What is the distance? T1 and D2. One. Okay, T1 and R4. Three. T1 and T28. T27. T1 and L3. 2. And T1 and A15. 14. T1 and F7. 6. T1 and V6. 5. T1 and G8. 7. What is that sum now? 65. Okay, 65 divided by L is the total number of residues. For example, if protein has 100 residues, okay, L equal to 100, and N is the total number of contacts. If that 100 residues means approximately it will have 400 contacts, right? Then you can see 65 divided by 100 into 400, that is the contact order for particular residue. But for a particular protein, this is for one residue. Likewise, do for all the residues. Then the number will be very high. Okay, yeah, because this is one residue, calculate for all the residues, and then divided by length is the number of residues. If it is 100 residue protein, it is 100, and N is the number of contacts. That means what is the total number of contacts each residue can make in a protein structure. For example, if you take 400, 500, then you can calculate this information. That is your contact order, right? If the contact order is less or more, if there are more number of long range contacts. For example, if you can see one protein has many long range contacts, one protein has only less long range contacts. Which one has more contact order? Because contact order depends on delta Sij. Long range contacts means they are very far away, right? If the protein has more number of long range contacts, automatically this SO will be very high, right? And less long range contact, this will be less. Accordingly, this is related to the folding rates. If your protein has to make more number of long range contacts, then it will take more time to fold. This is what the relationship between contact order as well as the folding rates. Okay, this is one concept. Then I'll show another concept. This is long range order. This is developed by our group. So here we can have the values LRO. This Nij by N, where Nij we define not here. Here they put delta Sij. Here Nij we define if I minus j is greater than or equal to 12. Right? So you can see all the contacts, right? So then if it is more than or equal to 12, then you can count it, otherwise it is 0. Right? How many contacts which are more than 12 here? What is Nij value here? Right? This distance between T1 and D2, it is 1. Is it more than 12? No, right? How many contacts, right? Where we have the difference of more than 12? Two cases, right? Because if you see the T1 and A15 and T1 and D28, right? So they have more number of the distance is more than 12. So here Nij equal to 12. N is the total number of residues in your protein, again normalized by N. So you can divide it, then you will get the LRO of particular residue. To get the LRO of the full protein, right? You need to get this number Nij for all the residues. For each residue, right? They have to calculate the contacts and how many contacts which are more than 12 residues, and then that will give you this Nij, and divided by this total number of residues, this will give you the LRO. So even this LRO is very high if the long range contacts are more, right? So it is less if the long range contacts are less. For example, if your residue has no only short range contact, this will be 0. Depending upon the long range contacts, you will have the numbers, right? Fine. This is another concept. Okay, this also have good correlation with protein folding rates. How your protein can take more time or less time to fold. With more number of long range contacts, it take more time, right? So to have all these contacts, right? Fine. So I got one more concept. Okay, I'll stop here. Maybe another 5 minutes. Okay, I'll do 12. Okay, another 3 minutes. I'll finish this. Okay. So yesterday also someone asked about the surrounding hydrophobicity, how to get this computational scale. Okay, I'll tell you how to get that. So for each residue here, right? So we can see the residues which are occurring within the limit of 8 angstrom. 8 angstrom we defined based on some analysis, but you can change the numbers. But this gave very good results. That is the reason why we use 8 angstrom. So for any central residue T, right, get the residues which are occurring within the limit of 8 angstrom, and use the values, experimental values. Yesterday I showed about Stanford scale experimental values of these numbers, right, and sum up that will give you the value of this t, right? So this value of this t depends upon all the residues which are surrounded by this t. There is another t here. So these both t's will have same values or different values. So my question is, this t will have some value and this t will have another value. So these two both have the same value or different value. Different, right? Because this t depends upon the residues which are surrounded by here, and this t we have to construct another sphere of 8 angstrom, and what are the residues which are occurring within that around this t, that will decide the value of this t, right? So okay, so if you take this t, what is the surrounding hydrophobicity now? You take the values from here, and can you calculate the value for this t? Anybody got the number? Okay, what is the value of this d? You can use this table here. What is the value of d? -2 or -2, right? So you can sum up all the numbers. Then can you tell the final answer? Can you get the number? What is the final answer? This -2 over the t. Okay, 2, correct, right? So this is -1, here 2, 2, 2, 2, so 8. This is 1, 2, 2, 2, right? 2, 4, 6, 7. 7 minus 5, that equal 2, right? So you can get this number. This is 2 is the hydrophobicity of this t. So take this example. I have another example here. What is the value of this t? So here it is surrounded with the d, r, l, s, k, t. Right? Now what is the value of this t? Can you calculate and tell me the answer? What is the value of this d? -2 r -2 l 2 s -1 k -2 t -1. So now what is the total value? What is the total value? -2 -2 -4 -2 -3 -5 -6, right? -5, correct, right? Okay. So now you minus 6. Now if you see this t, it has a value of plus 2. This t, it has a value of minus 6. That means both are trionic, but they are depending upon the location, depending upon the environment, right? So the hydrophobic character changes. That is the concept of surrounding hydrophobicity. It's not based on the single residue's hydrophobicity. It's based on the residues which are occurring surrounded by that particular residue. That's called a surrounding hydrophobicity, right? That's fine. So I'll stop here. So what are the different aspects we studied today? Right? What are different aspects we studied?
Uh, Sir.
Yeah. Yes. Right. We talked about the 3D structures, putting 3D structures, right? Yeah. Structure determination. And
Thank you for this. Yeah, thank you for this session, sir. Okay, let the question session continue. Yeah, participants, you can ask the questions.
yeah, what is the database for the protein structures? PDB, right? We discussed about the which tool we discussed about the visualization. Spymol. Then we discussed about the contact map and what are the various features we derived today from 3D structural coordinates, thermal accessibility and contact order, long range order as well as hydrophobicity, right? Okay. So now, yeah, if you have any questions, just feel free to ask and we'll discuss. This afternoon we have the hands-on on PyMod. Good, yeah. Research direction. There are so many ways to do the research in computer science. Maybe you can focus on any of these NGS analysis, or you can focus on the large scale big data analysis. There are a lot of options for the computer science students for the bioinformatics. The data science is a big boom now. So easily they can contribute a lot. Because I got two students from computer science. Yeah, they are doing the large scale data analysis. Any other questions? Okay, if there's no questions, then we will see in the afternoon regarding the hands-on session.
Yeah.
Okay.
Yes, participants, I think there are no more questions. Sir, once again, I thank our expert, Professor Gromiha, sir. I would request Dr. Preeti Sudha, madam, to thank our expert and participants. Ma'am, if you are please. Yes, ma'am.
So first of all, our thank you to the expert sir for giving us so much of time and from IIT Madras joining us from IIT Madras. And thank you so much, sir, for joining with us. And also, I want to congratulate Banav sir for organizing such a thing for us. Thank you so much, sir. Thank you to the participants also.
Yes, sir. Thank you, sir. Thank you for this wonderful session. So, sir will be giving his presentation during the afternoon. So, we look forward to that. And
yeah, I remind you to download the software, install the software PyMod. Yeah. Okay, I asked all the participants to install PyMod in your computer. Okay, please carry on.
Yeah. Yeah, sir. Thank you for this session. Yeah. So thank you, participants. Do join us at 1:45 for the next session. I thank our chair of session chat too. Thank you, ma'am. Thank you for coming.
Thank you, sir.
Yeah. Yeah. Thank you, ma'am.
Okay.